We are ready for a great victory over the mighty mouse. The cell membrane is our only barrier to get inside, but get inside we shall. It is made of a phospholipid bilayer. The phospholipid fatty acid tails are scared of the water, but the phosphate heads adore it. The tails hide by banding together, as we are now, and seek protection of the heads. Such weaklings. Cholesterol helps the membrane stay solid in high temperatures. He does this by not letting the phospholipids move as much. In the phospholipid bilayer, lots of proteins are buried. The lucky ones sit on the bilayer surface. Some of the proteins have glycoproteins and glycolipids attached to them. They act as antigens, allowing other cells to recognize them as the good guy. I have met some of them. The buried proteins carry substances through the membrane, since charged and polar molecules can't go through on their own. The tails kick them out. Luckily for Mr. O, he can get through on his own. And he gets to travel with his friends, the nutrients and the wastes. Mr. O's best friend, H2O, has to go through an aquaporin to enter the cell. Aquaporins only let water go through them. Every good barrier needs support, so we'll have to tamper with them to get in. The cell's support is called the cytoskeleton, which is constantly being taken apart and put back together. The cytoskeleton is made up of 3 parts: the microtubules, microfilaments, and intermediate filaments. The microtubules are the thickest of the bunch, and have a job of holding the cell together and moving. They help move the organelles, who we'll meet later, and help move the chromosomes when the cell divides. Microtubules also have the hefty task of moving cells, but they have some help from the microfilaments. Microfilaments, along with helping the microtubules, have a job of working the cell's shape. The microfilaments, made of actin, change the shape of the cell and cause contractions and relaxation in muscle cells. They also help out the microtubules in cell division by helping the new cell and the old cell split. Intermediate filaments anchor the nucleus and help shape it. All of the cytoskeleton's parts have different jobs to do, but they all help shape the cell, and without them, the cell would be weak and defenseless, which would be perfect for our invasion. But alas, it is not so. Within the animal cell is the cytosol, the great expanse of jelly-like fluid which we must cross in order for us to make our invasion successful. The cytosol is the fluid in which nutrients, like Mr. O, organelles, proteins, and enzymes float around until they are used by an organelle. This cell which we are invading is a prokaryotic cell. This means that its cytosol is called cytoplasm. Now, my siblings, swim to cell domination! The centrosome is where the microtubules are made, and it is also called the microtubule organization center. The microtubules help the structure of the cell by reinforcing it and making it squish resistant. Animal cells, like this one, have a pair of centrioles within the centrosome. Each centriole is made of 9 sets of 3 microtubules. These 9 sets are organized so that they are ring-like. During the process of cell division in animal cells, the centrioles replicate. Ahead I see the mitochondrion of this cell. This organelle converts ADP into ATP by extracting energy from sugars, fats, and other fuels with the help of Mr. O in both animals and plant cells. The number of mitochondria in the cell is determined by the cell's level of metabolic activity. Mitochondria consist of 2 separate membranes. The outer membrane is smooth and helps keep out unnecessary molecules. The inner membrane is highly convoluted. These folds in the inner membrane are called cristae. The space between the inner and outer membranes is called the intermembrane space, while the space inside the inner membrane is called the matrix. This matrix contains mitochondrial DNA, ribosomes, and enzymes which catalyze cellular respiration. Look ahead, soldiers. There lies the infamous lysosome, the membranous sac of hydrolytic enzymes which have been used to consume many of our mono brethren. It is a vacuole designed to break down macromolecules and used in phagocytosis. It was once thought that if the lysosome could be breached, then the cell would digest itself. However, the digestive enzymes in the lysosome work best in an acidic environment and do not work in the cytoplasm of an animal cell. The enzymes within the lysosome are made in the roughy R and changed inside the Golgi apparatus. The peroxisome is made up of a single membrane that contains enzymes. The enzymes take hydrogens from molecules and add them to Mr. O to form hydrogen peroxide. The peroxisome uses Mr. O to break down fatty acids and move them to the mitochondria. The peroxisome's main job is to detoxify poisons. It does this by taking the poison's hydrogen and adding exciting, Mr. O. Hydrogen peroxide is actually a toxin as well, but the cell is able to change the peroxide back into water if needed. The Golgi apparatus makes, stores, ships, and sorts proteins and other molecules. Products of any of the organelles are sent through the Golgi as materials, but most of the products that go through the Golgi are products of the endoplasmic reticulum. The Golgi apparatus produces a wide variety of polysaccharides as it modifies them in transit. The Golgi is made of a bunch of flattened sacs of membrane. When parts of the membrane begin to form a bubble and separate from the Golgi body, it is called a vesicle. The vesicles are sent out of the trans or sending side of the vesicle. The trans side faces away from the endoplasmic reticulum, while the cis or receiving side of the Golgi faces the endoplasmic reticulum. The endoplasmic reticulum is an organelle which has two different sections which are not separated by a membrane. The smooth ER has various functions based on the type of cell which contains it. In some cells, it helps to create enzymes which aid in the detoxification of toxins like drugs by adding a hydroxyl functional group, which makes them more soluble in water so that they can be removed from the cell. It also helps store calcium ions. The rough ER secretes proteins that are made by ribosomes. A ribosome is a complex composed of two proteins that make other proteins by translating mRNA. When a ribosome translates mRNA, it links together the amino acid sequence specified to make the polypeptides, or the components of proteins. The rough ER secretes proteins. Each protein is made by a ribosome that specializes in a particular protein. After the proteins are made, the ER membrane keeps them separate from the proteins produced by the free ribosomes, the ribosomes that are not attached to the ER. The ER transfers them through the transitional ER and sends them into the vesicles to the Golgi. A wild nucleus has appeared. This is the center of the cell and our destination where we must attack and insert our RNA so that the cell will replicate our genome. The nuclear envelope is the only obstacle that is still in our way. It is a double membrane which is perforated with pores. These pores are lined with a pore complex, which makes the inner and outer membranes continuous with each other so that the mRNA and the molecules can pass through. Everywhere on the nuclear envelope except the pores are covered with the nuclear lamella, a net-like array of protein filaments which allows the nucleus to maintain its shape. Within the nuclear envelope are the chromosomes. The chromosomes carry the genetic information of the cell. They are made of chromatin, a complex of DNA and proteins which close up into a thick mass during cell reproduction. In the very center of the nucleus is the nucleolus, where the mRNA is made. In order to make mRNA, the nucleus brings in proteins from the cytoplasm. Now, the cell is ours.
The cell wall is the first obstacle which must be overcome if this one is to invade. It serves as a protection from mechanical damage and aids in maintaining shape. These strong barriers aid the plant in standing upright. It is composed of cellulose, polysaccharides, and proteins. The cellulose is synthesized by cellulose synthase and is secreted into extracellular space, where it becomes embedded in the matrix of polysaccharides and proteins. Young plant cells secrete primary cell walls, which are thin and very flexible. These actively growing cell walls have cellulose fibers growing perpendicular to the direction of growth. Between the primary cell walls of adjacent cells is the middle lamella, a thin layer which is rich in pectin. This glues the cells together so that the cells can support one another. Certain cells also contain a secondary cell wall between the primary cell wall and the plasma membrane. This wall adds structural integrity to the cells which contain it. Even with these strong walls, they contain pores called plasmodesmata, which this one can use to gain entry into the cell. They are designed to allow water, small solutes, and cytosol from adjacent cells to pass through and spread nutrients between cells. Plant cells and animal cells contain many of the same organelles. One which they both have, but varies slightly between the two, is the centrosome. In animal cells, the centrosome contains centrioles. This is not the case for plant cells. The centrosomes in plant cells do not contain centrioles. However, they still serve as the organizational center for the microtubules of the cell. The chloroplasts are the organelles in which plants perform photosynthesis. The chloroplasts aid in the conversion of water and carbon dioxide into chemical energy by using solar energy. The chloroplast possesses three membranes, all of which are phospholipid bilayers. The first two membranes separate the cytosol from the stroma and the other contents of the chloroplast. These two membranes are separated from each other by a small intermembrane space. Inside of the two membranes is another membrane which is shaped like flattened sacs on top of each other. These interconnected sacs are called thylakoids, and their respective membrane is the thylakoid membrane. A sac of thylakoids is called a granum. The grana are surrounded by the stroma, which contains chloroplastic DNA, ribosomes, and enzymes. Thus, the chloroplast is separated into the intermembrane space, the stroma, and the thylakoid space. This one will require the energy created by the chloroplast in order that this one might replicate its RNA and create new TMV. Inside of both plant and animal cells are vacuoles. The plant cell which this one is invading contains a central vacuole. The tonoplast is the membrane of the central vacuole. It forms by the combination of several smaller vacuoles from the endoplasmic reticulum and the Golgi apparatus. This one calls the content of the central vacuole sap. This sap contains many of the organic and inorganic molecules which this one will use to replicate its RNA. The central vacuole aids in the growth of plant cells by swelling with water and creating pressure on the cell wall, and it will help this one grow in numbers.
Well, here we are again. It's always such a pleasure. Remember when you tried to kill me twice? Oh, how we laughed and laughed. Except I wasn't laughing. Under the circumstances, I've been shockingly nice.